Hello. Today we cover the cell theory and its historical background. Our knowledge about the cell started when we began to make lens implements that extended our visual perception beyond the macroscopic dimension we are normally accustomed to. No biological principle in history is as intimately associated with a tool than what the cell is to the microscope. In fact, it was the development of the microscope that extended our ability to see the details of living tissues and showed the microscopic realm of life. This led to the development of the cell theory. Since there is no universally accepted definition of life, the cell theory is the first major generalization that allowed us to get a better understanding of life. The cell theory, in the modern times, states that living organisms are made up of cells, that they are the basic structural or organizational unit of all organisms, and that all cells come from pre-existing cells. Today, we take these tenets or principles of the cell theory as a matter of fact, with no question or doubt. But the beginnings of the cell theory actually began with an old technology in the 17th century, that is of grinding glasses for eyeglasses or spectacles and magnifying lenses. It started with men who were skilled in lens making and had uncanny inclinations towards looking at extremely close and magnified surfaces of ordinary objects such as textile or cloth. The development of the cell theory owes its technological beginnings with a lens maker named Zacharias Janssen of the Netherlands in the 1600s. In spite of dubious reputation as a counterfeiter in the Netherlands, Janssen is posthumously associated with the invention of the compound microscope. Early in his career, Janssen was formerly employed as a guardian of two children of a well-known lens maker, Louis Louisen, the lens maker. And story goes that he took Louisen's lens-making tools and learned the trade of his former employer. And as a lens maker, he is said to have invented both the first optical telescope and compound microscope between the years 1590 to 1655. His purported invention of the compound microscope was his main contribution to the development of the cell theory. But according to popular idea, the microscope was really invented not for seeing microscopic organisms, but was rather used for examining the quality of textile or clothes, which was the important commerce of that time. After the invention of the microscope, people of other walks of life got the hang of it. There was Reverend Athanasius Kircher, S.J., a German Jesuit scholar and polymath, who took a notably modern approach to the study of diseases, especially of the pandemic of his time, the Black Death or bubonic plague. As early as 1646, he used a microscope to investigate the blood of plague victims. In his book Scrutinium Pestis in 1658, he noted the presence of little worms or animalcules in the blood and concluded that the disease was caused by microorganisms. His conclusion was correct, although it is likely that what he saw were in fact red or white blood cells and not the plague agent Yersinia pestis. He also proposed hygienic measures to prevent the spread of diseases, such as isolation, quarantine, burning clothes of deceased persons, and the wearing of face masks to prevent the inhalation of germs. More and more, natural philosophers, the old name of biologists in pre-modern times, got hold of the microscope. A Dutch biologist and microscopist, Jan Swammerdam, was considered the most accurate of classical microscopists, who was the first to observe and describe red blood cells, at the same year as Father Kircher, S.J. did in 1658. He was the first to observe under the microscope that a frog embryo consisted of globular particles, alluding to the presence of cells that comprised the developing embryo. Jan Swammerdam was one of the first people to use the microscope in dissections when he studied stages of insect development. But it is Robert Hooke who stands as the discoverer of the cell during the development of the cell theory, because he was the first to formally describe and gave the name of the cell in his book, Micrographia, in 1665. The discovery of the cell is attributed to him. Micrographia is the first book that illustrated insects, plants, and other organisms as seen through the microscope. But it's funny to note that when Hooke described cells from cork tissue, what he actually observed under the microscope and described to the Royal Society of London were dead and hollow cells of the cork tissue, the outer bark of mature trees. Nevertheless, the term has stuck for good, even up to this point in time. We use the name cell. Particularly best at lens making, a clothing merchant by the name of Anthony van Leeuwenhoek in the 1670s explored not only the quality of his textile, but most notably microbial life with his own single-lens microscope. Although he never wrote any book, his discoveries of cells and other microorganisms were documented through his correspondence to the Royal Society of London, which published his letters. Considered the father of microbiology, Leeuwenhoek was the first to experiment with microbes and first to document microscopic observations of muscle fibers, bacteria, sperm cells, red blood cells, even crystals in gouts, and blood flow in capillaries. Strangely, however, it took about 200 more years for the historical development of the cell theory to catch up. In 1833, Robert Brown, a Scottish botanist and paleontologist who was then studying the leaves of orchids under a microscope, discovered the nucleus as a regular feature in plant cells. Brown published detailed description of the cell nucleus and cytoplasmic streaming in 1833. The cell theory was finally declared 5 years later by Schleiden and Schwann in 1838. They both declared that the cell was the basic unit of plants and animals. Matthias Schleiden, a botanist, and Theodor Schwann, a physiologist and physician, published their findings almost simultaneously that lay down three main tenets of the cell theory, stating that all plant and animal tissues are made up of cells, and that the cell is the basic unit of structure and organization in organisms. 22 years later, in 1855, the German physician Rudolf Virchow added the third principle of the cell theory when he discovered that new cells formed out of the division of cells. Thus he introduced the third important dictum of the cell theory, stating omnis cellula et cellula, meaning all cells arise from pre-existing cells. Virchow is also best known for introducing the idea that diseases occur at the cellular level, laying down the foundation of cell pathology and the fundamentality of the cell concept. After the publication and establishment of the cell theory, discoveries of other features of the cell, that is, cell organelles, also quickly followed. In 1888, Albert von Kolliker, a Swiss anatomist and foremost histologist, identified and described the mitochondrion. Camillo Golgi described the Golgi apparatus in 1898. Keith Porter discovered the endoplasmic reticulum in 1945, while Christian Ledoux discovered the lysosomes in 1955 and the peroxisomes in 1965. Ernst Ruska and Max Nutt built the world's first electron microscope in 1931. The electron microscope employed electron beams as illuminating medium and magnets as magnifying lens to enlarge images of the minutest detail of the cell a million times over. It therefore allowed us to see ultrastructures of the cell that would have been very hard to visualize under the ordinary light microscope. Please pause this slide for a while to take note of the assignment for this lesson and upload it in the eLearn Upload Hub designated for this assignment not later than December 18, 12 p.m. With the rapid development of tools and techniques in cell biology in the mid-20th century, research on the cell as the structural and functional unit of life reached to a frenzied activity in the 1950s. Everyone Everybody wanted to know more about the cell with the available tools of cell biology. From this research explosion, the modern tenets of the cell theory were upgraded, which are stated as follows: that all known living things are made up of cells and their products. The cell is the structural and functional unit of all living things. All cells come from pre-existing cells by cell division. Cells maintain hereditary information, which is passed from cell to cell during cell division. All cells are basically the same in chemical composition. All energy flow, that is metabolism and biochemistry of life, occurs within the cells. Next meeting, we talk about the cell structures or organelles and their functions. Thank you.